This line graph illustrates a clinical symptom known as accelerated long-term forgetting, or ALF. Patients encode new information normally, performing perfectly on immediate recall tests, but at the 30-minute mark, their retention curve collapses. Over the following days and weeks, the acquired information is almost entirely lost. Historically, the consensus attributed this rapid loss to structural hippocampal damage or a failure in late-stage memory consolidation. If a memory could not be consolidated, it simply decayed. However, clinical data renders that explanation insufficient. ALF routinely appears in populations with Alzheimer's, Parkinson's, and certain forms of epilepsy. Many of these patients possess structurally intact hippocampi and show no seizure activity during the memory retention interval. The physical hardware for consolidation remains present and active, yet the memory still vanishes. The persistence of ALF in these patients suggests that the brain does not simply let unused information fade away. Instead, it operates intrinsic parallel mechanisms dedicated entirely to erasing information. Neurodegenerative memory loss occurs when highly active, targeted erasure mechanisms override the brain's consolidation efforts, forcing the removal of otherwise stable information. Memory persistence relies on a push-pull architecture. The forces of encoding and consolidation are constantly competing against the forces of intrinsic active forgetting. This continuum maps clinical memory disorders based on that structural competition. On the far left, we find conditions defined by hyperactive erasure. In Alzheimer's and Parkinson's disease, the mechanisms responsible for dismantling memory traces, or engrams, operate at an accelerated pathological rate, producing the symptoms of ALF. The opposite pathological extreme exists on the far right. Conditions like post-traumatic stress disorder or highly superior autobiographical memory represent a state of hypoactive erasure. Here, the brain's natural forgetting machinery fails to engage, leaving information permanently accessible. This shared continuum implies a common regulatory mechanism, one capable of being modulated to address either pathological forgetting or pathological retention. In order to successfully treat these debilitating disorders, clinicians must first identify the specific molecular agents that actively dismantle the engram. Moving from the clinical scale to the cellular scale, we can observe exactly how memory traces are physically dismantled. During rapid consolidation, active synapses in the hippocampus form tight physical connections, synthesizing new proteins to maintain the memory structure. This diagram highlights an engram's postsynaptic membrane, specifically GLU-A2 AMPA receptors. Erasure initiates here via NMDA activity. Through endocytosis, GLU-A2 receptors are pulled internally, physically weakening the connection. Finally, non-neuronal microglia surround and physically prune the remaining synapse, executing a permanent structural elimination. This microglial pruning does not happen instantly. It operates on a delayed timeframe, running continuously in parallel to systemic consolidation processes. Forgetting is an active form of neuroplasticity. It requires energy, biological resources, and continuous physical circuit remodeling to keep the brain's networks efficient. Controlling this physical erasure requires intracellular signaling pathways capable of changing the actual shape and size of the synapse from the inside out. Inside the dendritic spine, the structural scaffolding is made of an actin cytoskeleton. The dynamic remolding of this actin network is regulated by a signaling protein called RAC1. To function adaptively, a receptor-based mechanism tells RAC1 when to alter the spine's morphology. When dopamine binds to these receptors, it triggers RAC1 to restructure the internal actin network. This visibly shrinks the dendritic spine, physically degrading the memory trace. Dopamine signaling facilitates both learning and forgetting, often triggered by a mismatch between expectation and reality. Beyond its role in reward, dopamine serves as the biological trigger that instructs the RAC1 pathway to begin remodeling the synapse. Applying the RAC1 hardware model directly to clinical pathology explains the onset of accelerated long-term forgetting. This matrix maps disease states to pathway activity. In Alzheimer's, elevated amyloid beta chronically activates RAC1, accelerating erasure. Similarly, Parkinson's neurodegeneration disrupts dopaminergic signaling, triggering the forgetting cascade. Contrast this with PTSD. Blocked RAC1 and dopamine pathways trap the engram in a persistent state, preventing pruning. Clinical memory disorders are specific mechanical imbalances in the RAC1 and dopaminergic erasure pathways. If active erasure is the biological default, how do humans retain highly detailed, stressful episodic memories without them being pruned? A study by Zhang and colleagues tested this override mechanism using the Trier social stress test to induce acute cortisol spikes in participants. This flowchart outlines the behavioral pattern separation object task. Participants view a target object, and 24 hours later are tested with a highly similar lure object to measure precision. Success requires the dentate gyrus to create distinct representations, known as pattern separation. The researchers found a critical temporal distinction. When acute stress was induced directly before retrieval, it had no significant effect, but when induced during early consolidation, results inverted. A linear regression graph shows a positive correlation between cortisol spikes and correct discrimination of similar stimuli. Cortisol specifically modulates the dentate gyrus, heavily enhancing its ability to execute pattern separation during the encoding of the event. The evidence suggests that an acute cortisol spike during early consolidation fortifies the dentate gyrus, shielding the engram from baseline RAC1 erasure. This scale visualizes the competition at every synapse: consolidation by the dentate gyrus versus erasure by RAC1 and microglia. Under stress, cortisol tips the scale toward consolidation, locking the engram into place. When amyloid beta spikes, RAC1 forcibly tips the scale to erasure, dismantling the engram. Accelerated long-term forgetting is the clinical manifestation of this scale tipping permanently to the right, neutralizing information regardless of how strong the initial encoding was. Retention relies on a volatile biological standoff, requiring the constant active maintenance of the engram against the brain's inherent drive to erase it. Abandoning the passive decay myth carries direct pharmacological implications. Treating ALF in early-stage Alzheimer's patients requires targeting the erasure side of the scale. Clinical research is currently investigating the true viability of RAC1 inhibitors to artificially suppress this overactive pruning mechanism. Conversely, treating pathological retention in PTSD requires targeting the opposite mechanism. Researchers are exploring dopaminergic agonists to manually trigger prediction error signaling, initiating the eraser sequence the brain failed to start on its own. These emerging therapies act as targeted counterweights, pushing against the disease-specific disruptions to return the patient to a state of adaptive forgetting. By understanding forgetting as a highly active, mechanical process, clinical neurology gains the exact biochemical coordinates required to modulate the persistence of human memory.
Thanks for watching. Make sure to like this video and share it with your friends. And don't forget to hit that subscribe button.